Hello, everyone, and welcome to today's webinar. My name is Nathan Nee, Senior Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speaker Andres Nevarez will define the triple-negative breast cancer morpholome and discuss morphological insights into TNBC progression and metastasis. Before we begin, I'd like to thank Deepcell for sponsoring today's webinar. We'd like our webinars to be interactive. We encourage you to send us your questions or comments at any point, and our speakers will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsor has provided us with some helpful resources related to today's webinar, and we have posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you have minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This webinar will be archived on the Scientist website, and we will send you the link for the on-demand version via email within a couple of days. Please note that you will not be able to download the presentation slides. Before we begin, we have a short message from Deepcell, represented here today by Kelly Miller and Courtney Anderson. So I would just like to invite those two to please take it away.
Good morning, good afternoon, depending on where you're located. Thank you so much for joining us. I'm Kelly Miller with Deepcell, and it's my pleasure to briefly introduce our technology ahead of the sure to be exciting presentation from our friends at MD Anderson. At Deepcell, we're introducing a whole new concept of cell analysis and a new type of analyte. So I want to approach the topic by starting with the why. We're living in an age of single-cell biology where we're all approaching science in a more and more precise manner, aiming to gain more and more granular and also authentic glimpses of nature. And the variety and sophistication of technologies available today is impressive, to say the least, but we're still striving to address remaining obstacles that stand in our way of gaining a complete view of biology. As one set of examples, and this is certainly not exhaustive, there are scenarios where trying to isolate or enrich a specific cell population isn't possible simply because there aren't markers available for that population. I mean, you can imagine using single-cell RNA-seq to identify a rare cell population that, for example, is maybe resistant to drug or is specific to your disease setting, but there are no markers available to isolate and characterize these cells. Or you know that a certain population of interest is particularly sensitive to perturbations like staining, so you want to avoid any labels. I mean, beyond these examples, it's safe to assume that among the vast continuum of cell types and cell states, we don't yet have the full picture using the current portfolio of omics tools, and we need to continue examining cells in new ways to get that holistic view. This is all not to mention that single-cell technologies still have significant time and cost barriers, which make faster, cheaper alternative technologies all the more attractive. And in fact, it's precisely some of those challenges that led to the development of our technology and the founding of Deepcell, specifically the need to characterize stem cells throughout differentiation and the need to screen gene-edited cells for disease-associated changes. To address some of these challenges, our technology leverages morphology-based analysis, which has long been recognized to hold significant information pertaining to cellular phenotype and function. And here you can see a variety of examples from the literature in which morphology provides key information, in this case about everything from cell state to differentiation status. and onto target identification and effector function, as well as cell health, which is classically defined by morphological criteria. So, if morphology is so information rich, why is this field behind and why don't we use it more often? And the reason for that lies in part in technological limitations. So, here you can see an example of two very different cell types. This difference is easily recognizable by eye, I think you would agree. Current tools allow us to provide a qualitative, at best semi-quantitative assessment of the morphology of these cells that is somewhat subjective and in most cases is very low throughput. What we need, on the other hand, is a measure that is truly quantitative, scalable, and robust, so that, for example, we can recognize functional differences like this. These two cells are from the same cell line. Exposure to drug has resulted in one of them becoming drug resistant while the other is drug sensitive. And in this instance, we lack antibody labels for this functional difference, and we lack the computational ability to discern these subtle differences by our own eye and by current methods. Deepcell, we're relying on a cutting-edge AI model integrated into our platform to extract high-dimensional single-cell morpholomics data. Our Human Foundation model, or HFM, is a self-supervised model that's been trained on millions of cells that acts as a feature extractor. And we're extracting two kinds of information using both a computer vision encoder, which extracts human interpretable features like cell shape or texture, and a neural network that extracts deep learning features. Together, the Human Foundation model extracts 115 quantifiable features of morphology, which turns morphology into morpholomics, a new quantitative high-dimensional ohm. The data that comes out of the foundation model can be depicted in many ways, and here we show a couple varieties of morphology UMAPs, depicting the data in a very similar way to how we typically visualize single-cell RNA-seq data, for example. Using this data, we can dive even deeper into the quantifiable nature and extract the differential features that underlie the separation of given cell populations or different morphotypes. Each differential feature is weighted by importance and can be quantified in density plots or a variety of different types of histograms and bar plots, as you can see on the right-hand side. The Human Foundation model is just one component, a key component of our overall platform that's integrated with our REMI instrument and our Axon data suite, which together encompass an end-to-end solution that includes single-cell high-resolution imaging and real-time characterization. This characterization comes along with the ability to sort cells that have similar morphotypes or morphologies into up to 6 different output wells so that you can use those then for downstream analyses. And here you can see an actual picture of the REMI instrument as well as our cloud software interface. So with the help of this technology, we are striving to support scientists to elevate morphology and turn it into a quantitative, robust analyte. Just as we've seen genomics and proteomics, for example, rapidly advance over the last decades, we aim to do the same with morphology. And the idea here is that doing so will allow us to capture cell biology more holistically. I want to thank you all for your attention today. This is the last slide for me. If you're interested in exploring our data, please check out our demo data sets on our website. And for more information, please visit us at deepcell.com or follow us on LinkedIn. Thank you all again.
Thank you very much. With that, let me introduce our speaker. Andres Nevarez is a postdoctoral fellow at the University of Texas MD Anderson Cancer Center. He completed his PhD in biology at the University of California, San Diego, where he studied the intersection of cancer biology and artificial intelligence through quantitative cell imaging. In his current research, Nevarez investigates cancer metastasis through a morphology first imaging approach that captures rare cell physiologies and identifies disease state properties that sequencing first approaches may miss. Let's just make sure everything is up and running from a technical standpoint.
Yep. Alrighty.
There
Is
we go.
that is everything here?
Looks like it on my end. Andres, please take it away.
Alrighty. Thank you for the opportunity to talk today. Really excited talking about the early experiments in this project of defining the morpholome of triple negative breast cancer and expanding it across the different stages of breast cancer. I'm in the NAVIN laboratory at MD Anderson. And here I have in the background is an indigenous Mexican folk art style. And I asked Dolly 3 to generate some images in the style of what the morpholome is. And, we just got done talking about this new field, and there's only very few papers like out there that are using the term morpholome. And it was really interesting to see taking these papers into account as data sources that you're looking at, intracellular organelles, cell shape, cell size, and a lot of things that may not even be human interpretable. I just thought it was really cool and wanted to share this as a background and an intro for our presentation today. So first, I need to disclose I'm a Deepcell scientific contest winner. I'll get into that. I have no interest or stake in the company, and I'm presenting the data we generated from the grant and not the representing UT MD Anderson's Cancer Center views on the product. So the old adage is seeing is believing. And, just over 100 years ago, Ramon Cajal and Golgi had won the Nobel Prize for being able to stain and see single neurons. And he was able to classify this and look at the different morphology and drew amazing illustrations of the different morphologies. And he was able to get some qualitative insight into how the neurons and their morphologies relate to cell state and function, which is allowing us to, imaging is allowing us to observe and track how cells behave. Their appearance and behavior is really important and provides opportunities for us to learn about the cell's function, because morphology is directly related to cell state. You can even think at it as a systems approach. It's a very abstract approach to be able to understand the cell state. And really, the morpholome is an emergent property of this complex genomic gene and expression changes that happen in disease progression of cancer cells. So we've talked a little bit about what the morpholome is and these quantifiable features observable through imaging-based technologies. Wanted to plug my recent paper looking at the non-coding mutations effect on the morpholome. But the way I like to see it is it encompasses cellular and nuclear morphology. I feel that's the obvious part. But we can get dynamics and lineage analysis in there of looking at morphodynamics. Depending on how you collect your cells, you can get some information about translocation and motility, and even lineage analysis, as some of the data I'm going to show today can all be incorporated, as well as cellular organelles. I know we're using bright field data, but even a few years ago, the Allen Institute was able to use bright field data and gain information about intracellular organelles and other things using AI models. And that's something I'll discuss at the end of future directions of where we want to be taking this, the data that we get out of the Deepcell platform. And ideally, you are able to integrate some of these features and to be able to select these for downstream single cell sequencing. Our lab is a pioneer of single cell sequencing, and we have a lot of novel single cell sequencing technologies that can allow us to do co-assays, probing the genome, the epigenome, and the transcriptome all from a single cell. And so we need to be able to get our cells in a format to do this. So we have this classic Waddington landscape, but I've amended it a bit to look at the metastatic state and the cell appearance. And so the morpholome, again, is an emerging as a critical lens through which we understand these intricacies. And because the disease landscape of cancer can be so convoluted, there's been a limitation of single cell genomics and even a limitation of personalized medicine, in that it hasn't really worked. And talking to a lot of different cancer centers, we've been able to gain some insight, but it hasn't come to fruition as we had hoped. And that's where we can take a phenotypic to genotypic approach, or studying the morpholome and relating it to the underlying signaling networks and understanding how the genetic heterogeneity is influencing this. And it's not a passive reflection, but offers these deep insights into characteristics such as, the cell cycle, its tendencies to adhere to structures, responsiveness to drug treatments, even its relative lifespan or specific gene expression patterns. And importantly for us, or what we're studying is the metastatic stage, its potential to invade surrounding tissues. I like to think of it as an observable outcome that encapsulates this delicate dance between the cell's biochemistry and its physical form, and to really understand and connect this rich omic information together to build these multi-omic models to understand how cells metastasize and why they're able to overcome stressors of going and invading in the body and able to colonize and thrive in a new organ that can be radically different than where it started. There's a review article published talking about quantitative lifestyle imaging approaches to studying the metastatic single cell state as well. I was having a conversation with one of my colleagues, and we were talking about this. A lot of people in the lab are doing single cell genomics or transcriptomics, and we have this biopsy sample. And we've come to this stage in the field where we have ohm relativity. So if we dissociate the sample and we get single cells, the morphologies can be different. And when we're taking all of these cells, typically when we do, our transcriptomics and single cell DNA sequencing, we could pull out like copy number aberrations. We can pull out, some of these cell states, some gene expression changes. But this is for a morpholomicist, this is a bulk experiment, because we're then putting all of these different morphologies together. Whereas for us to combine them into morphotypes and then sequence those morphotypes allows us to focus and actually do these single cell experiments. Now, from the other perspective for my colleagues is that this would be considered a bulk assay because you're pooling your cells together. But the technologies we have in our lab allows us to take these cells and individually, into a variety of techniques that we have, probe what's happening inside the cell. So it's still keeping it as a single cell assay. And as I'm depicting here, there's heterogeneity even within the morphologies in which we're able to understand at a deeper level once we connect this to its other omics. But how do you get from one ohm to another, especially with morphology? Oftentimes, you need to allow the cells to exist in a state that's appropriate for downstream single cell sequencing. You have tissue harvesting and single cell suspension, and here I'm depicting Deepcell from their landmark paper that came out of, as we talked about, a machine learning and deep learned based metrics to be able to sort. One of the things I want to say here is that long before what we've been doing before with traditional microscopy, right? Microscopy has been around for centuries. We've been using microscopy to understand morphology for a very long time, but the limitation has always been, how do you get cells at a high throughput way into a state that is appropriate for single cell sequencing? We've used laser capture microdissection, robotic arms that will pipette out little amounts of trypsin and be able to take these cells, but it's very low throughput and it's dependent upon the operator. And you're looking qualitatively at the cells and just trying to see gross differences and then take those cells and sequence them. But as I'm going to show later on, some of these machine learned human based algorithms, computer based, vision based algorithms, are not always the best way to go. While they're easily interpretable, it may not be the best way to classify the differences between these cells. And so I was lucky enough to partner with Deepcell through a 2023 Spring Science Challenge and to get some samples run by the company. And we've gotten through part of those samples, but it's really interesting to start off with some of the data today and hopefully follow up later on with even more of the samples that we've been running. And so it's been wonderful working with them, trying out the machine and their all the applications that go along with it. So there's some key definitions. I think this might be a general audience, and so this is just to orient us. So we have, a cell group. When you're looking at UMAP, you just are drawing like a circle, polygon around it. You have our embedding model, which for Deepcell is a human foundation model. You take a cell image, you turn it into numbers. You have the embedding, which is the numbers, and that's what the human foundation models outputting, and then you're able to show how these images are close together in this high dimensional space. We talked about morpholomics, we talked about morphotypes with these distinct morphologies, a UMAP being a projection model, and a bit about these importance scores. And when I get into explaining our different morphotype groups, I'll get more into how important this importance score is for us to understand what these features are, whether it's deep learned or computer vision based features, how we can interpret them and understand more about the biology. And so the workflow is we have our single cell suspensions once we get our cells, our tumors dissociated, and we are able to load these samples in the microfluidic chip. And then typically you're running about like 10,000 cells at first to develop your own multi-dimensional visualization. And so you're able to go through the imaging, your morphological analysis of the images, and then draw gates around your groups and then sort them out. And it's been working really great with the cloud-based data analysis. So the human foundation model, like we talked about, is trained upon label-free, assesses 115 dimensions, and it's been developed on millions and millions of cell images, and really needs to be thought of as a feature extractor. And so with the deep learning encoder, we mentioned this just a few minutes ago, but just to go over it again, we're looking at the human foundation model deep learned, feature 1 all the way up to 64. And then we have these human constructed algorithms like position, cell shape, texture, intensity, that is more readily understandable. And we're combining both of these for accuracy and interpretability for our morpholomic profiles of our samples. And what I like to think of is, this is a great image to show from a paper a few years ago about imaging cytometry from Broad, and it's called deepometry. And so when you're going through, you can segment your cells traditionally, and then you do all of your feature extractions, which ones are different, and then do your supervised learning. And with the deep learning, you can see these cells here in these images. And as you go through the layers, you're becoming more and more abstract. You see here's kind of the first pass of these cells. It's focusing on edges and certain ridges, different textures, it's more and more abstract and less interpretable as you go deeper into the convolutional neural net, which is what Deepcell, one of, not exactly what Deepcell uses, but in a general way, of a CNN. And so we're able to understand these and get these images, and things like area for cell cycle is really obvious, or different cell types. And we can understand that long ellipse axes. This can be a mesenchymal to epithelial transition, or vice versa. Looking at the position of where the nucleus is inside of the cell can give information about nuclear piston locomotion and movement. And so this is really a great way to understand if there's these large differences between cells. But oftentimes, we can get information about blebbing, vacuolization from this, but the deep learned models are much better and can focus on things that we can't see. And it's oftentimes the things that we can't see are best for classification to sort out these different populations. And so we have our deep learning and morphometrics. We normalize PCA trend, our classifier, and we have our UMAPs that everybody is familiar with. And so I'm going to be representing the data today as we show this Leiden UMAP, which is identifying clusters with distinct morphologies. And then we have our sample UMAP, which is just looking at the different samples, sample 1, 2, 3. And then a density-based UMAP, heat map, for where the majority of those cells lie in the overall UMAP, which gives a lot of great information to tell the differences between these samples. So this brings us to our question for this particular project is we wanted to really test the system. And instead of just looking at different cell types, which I feel would be pretty easy, I wanted to look at clonal enrichments and differences, single cell clones from the MDA MB231 triple negative breast cancer cell line. And in a 2021 paper, the Nevarez lab made these clones along with other cell lines. And we were looking at the copy number aberration differences. We were looking at transcriptomic, single cell transcriptomic, and genomic differences. And we found striking differences between them, genomic drift happening after a few passages. And copy number aberrations are changing in just a few passages there. And so we wanted to see if these copy number aberrations and changes with the transcriptome profiles, are there going to be differences at the morpholomic profile level? And we expect there to be differences for clonally distinct cells, so lineage analysis to have the most different, and then more similar clonal lines at the genomic and transcriptomic levels. Are there going to be distinct morpholomic differences? We looked at MCF10A, these normal breast cells, to determine if there's morpholomic trajectories or unique morphotypes. And we included the parental line to determine if the clonal heterogeneity is recapitulated in the clone, or is it within the realms of the parent at least, or is there differences in distributions of the different morphotypes within the morpholome. And so here we have our samples. I'm just going to call the clones C28, C28, C10, C1, C35. We have our parental and our MCF10. So here it's just based upon our samples, IDs. And you can see here that C1 is kind of occupying the space, which is really interesting that we're going to dive into. And then we have our Leiden, which is focusing on differences in morphotypes, so grouping them together. And we're seeing some groups overlapping with different samples. So let's dive into that. If we have our Leiden UMAP here, what I really like about the Deepcell system is that you can look at the images and hover over them and see, are there some observable differences, and kind of do some of these quality checks here and see that there's more texture, larger, some unique characteristics over here where the cells look more similar, but you'll see later on is these deep learned features are discriminating and able to uniquely classify these as different morphotypes. And so it gives us some insight, and that's really helpful. I've tried some of these other systems that are on the market of the BDSA with CellView and others in our traditional non-sorting image cytometry, and it's not as clear to be able to do this. It's the software and technologies. You can't really do something like this. So this is really a great way to investigate your data quickly. It's all within the Axon suite, and you're able to do this quickly. You don't have to do post-processing of it in order to gain some information, as you would have to do with the other systems. And this is just a montage to look at this for each one of our different light-in categories. And I want to note that these are not apoptotic blebbing. Deepcell has some literature out there on their website that looks at the apoptotic blebbing. This is not it. They're able to identify apoptotic blebbing and necrosis using the human foundation model, and they are discarded. It's not focused on that. And even if you want to investigate just looking not at just different cell types, but tumor cells within a mixed biopsy, you're able to do that as well using the human foundation model. And so this is a great way to do mixed analysis of samples, and you're able to have some confidence in what you're looking at, if it be a tumor cell or a non-dying cell, and in future looking at these cell cycles, so that way you can focus on cells that are even within the same cell cycle. Okay, so I'm going to be going over the important features. I just wanted to bring this up so we can reorient ourselves. So these important scores are determined by how much a random forest classifier depends on each feature. So you're going to get your features for your different light-in groups or samples, and it's determined using this random forest classifier of what is important. So when you remove, let's say, human foundation model deep learned feature 22, how does this affect its ability to classify and differentiate between populations? And what I think is really cool is within the software, again, this is a monocyte to macrophage example, but within the software, you're able to make these plots on the fly right away and can visualize your data just after you do your 10,000 cells, and then make calls about what you're going to use in order to separate your populations. So this brings us to ours. This is heavily dependent on the deep learned embeddings, which is not surprising, actually. These are nearly identical cells. They all come from the same parent, except for the MCF10 cells, but it's sharing a lot of the same breast lineage characteristics, right? And so we are able to look at these different groupings and to see how they're different. Only one as perimeter shows up of these classic human generated algorithms. And I think we saw that a bit when we were looking at these different groups of the montages, that there can be differences in the perimeter size. And so if we have these important deep learn embeddings and we have images over them, we can gain insight into it. But again, it may not be entirely possible because these may not be readily apparent by eye, but they're extremely important for us to create our gating. And so then focusing on these features allows us to at least gain some insight quickly, and then do our sorting based off that. And so if I want to look at, this deep learn embedding 48 to see how it is, and hopefully be able to classify, sort based upon that, and you do the single cell sequencing, you are getting information about what this deep learn embedding is, because you're connecting it and you're creating somewhat of a dictionary. You can even, if you want to, just take this whole group and just say they are defined by these embeddings. That's something you can do as well, and give insight of what the AI, really open up the black box of AI in a different way and do it quickly while you're doing your experiments, is extremely important. So we come to our density plots, our density UMAPs, and we're looking at our different samples, the normal breast MCF10A here and the 231 parental. And we're seeing a lot of interesting things. And so let's break this down. We see that we're looking at the light in here and the distributions. It's just plotted differently. So you're seeing C1 with this heat map. It's pretty localized to this light in group 3. And hovering over it, it does show a lot of these unique morphotypes that you can see a bit. And the distribution for that clone is very limited. I mean, it has some in this arm here and spread out, but from looking at the parental, it's a small group in the parental, and it's not really occupied. If you're looking here, it's occupied at a much, much lower percentage. And so when these cells are created in, randomly by serial dilution, and then you're able to take this population, it's showing that these morphotypes are conserved and they are a tight group, similar to what this Waddington landscape, the hypothesis originally is that you have this large genomic heterogeneity that's affected by so many different things. But you would hope in the hypothesis is that it condenses on a smaller number of morphotypes and morphologies, and we are seeing that even with C28 here. It's being heavily a lot of percentage of cells are in this group 1. And we are seeing that there's unique morphotypes even for our MCF10A. So these normal breast cells contain a unique morphotype seen here. And these hotspots are different compared to the 231 parental. 231 parentals pretty much spread out with a hotspot close over here, but the hotspot is not really recapitulated in the other clones. It's similar to the C1, but at a lesser degree. But then the MCF10As are having these morphotypes that are not even present in any of the clones, definitely to a much less degree, I should say. Not that they're not present, it's to a low percentage. But the parental is occupying this area at a higher percentage than the clones, and you're seeing that there is some conserved, like this morphological trajectories in these morphotypes that go from a normal breast to a triple negative breast cancer cell. And that the clonal heterogeneity exists within the parental cell line. So it's not an entire recapitulation as you might think. The distributions are not the same for the clones compared to the parent. They occupy space, but it's not identical. So it's like altogether they're recapitulating this space, but to different hotspots, which I was not expecting. I thought there would be, more of a not as many hotspots and more of a smear across these density UMAPs. And so this was really interesting, and to see that these distinct clonal cells that have their own copy number aberrations and their own genomic and transcriptomic profiles are condensing on unique morphotypes that are not present to the same percentage in the parental. And we're seeing this in comparing the parental in these different groups, right? So if you're looking at the normal cells here and in this area, you see that cells here in the normal and this clone C10, they're occupying a similar space, but albeit different. And they look similar, but the deep learn embeddings are different. So it's picking up on these subtle differences, and you can kind of glean what it would be. There's similarity in the texture here, whereas here there seems to be a little bit more polarization, some bodies happening. And then this unique morphotype for the MCF10A, you're having what looks like to be a little bit larger cells, some dark connected pixels here, so looking at intensity. These things relate to physiology, and it's relating to, since it's bright field, it's labor free, you're looking at light absorption, light scattering. Features like that have been used by pathologists for a long, long time. So just these generic features have been used. And I like to think of this assay as like live cell pathology. And to how do you investigate its physiology using the pathology of what's going on and to understand this disease progression at different states. Instead of looking at clones, you can imagine looking at a normal breast sample to DCIS to invasion, and to understand how the morpholomic profiles evolve over time, because we're already seeing that there's distinctions and distribution changes happening at the clonal level of just the tumors, and we know there's clonal enrichments that happen in for this progression. So how do these different clonal expansions of these cells affect the fitness and affect which location it's going to in the body, and where is it metastasizing, and how is it able to thrive in these distinct organs and environments despite therapeutic interventions. So in summary, we image these 4 clones, the parental normal cells and samples, and we see that it contains the normal breast contains a unique morphotype. The clonal heterogeneity exists within the parental cell line, albeit not at the same distribution. And this platform offers the Deepcell platform offers a conducive product that allows us to do single cell sequencing and downstream analysis. Now, I didn't go into what these profiles are. The data was published, but we're reworking some of the data to focus on just the clones and looking at different morphodynamics over time with these cells as they progress. And so that will be coming out. We're preparing a manuscript for later this year. And so this is already shoring up to be a really interesting story to create a real connection between phenotype and genotype at the single cell level. And so that's what we've been striving for in the Navy lab to do. And it's actually a paradigm shift in our lab, whereas we've been focused on the genomics and sequencing technologies first. And this is a completely different area of going top down first and seeing what metaprograms are enriched when you're looking at looking at single cell transcriptomics. What metaprograms are enriched? How does the copy number change, and how do these clonal events and clonal expansions relate to different morphologies? So I obviously want to thank all of the NAVEN Lab and the Deepcell grant, and I'm happy to take any questions about this and any future prospects out there.
Thank you very much, Andres, for that presentation. So we're going to start our Q&A session now, and just as a kind of a final reminder to our audience that they can still submit any questions that they might have during the Q&A session, and we'll try to get to as many as possible. Okay. So the first question I want to ask, and I think the question itself asks whether you can differentiate between macrophage populations. So the example requested was M1-like versus M2-like. I think this maybe extrapolates a little bit further in terms of how do morpholome clustering and morpholome phenotypes, how do those reconcile with what we already know in terms of like delineating or separating cells by genotype or by other phenotype cell surface markers, something like that? How does the morpholome data match with what's kind of already established when it comes to characterizing various cell types?
Yeah, I mean, from the data I've seen out of Deepcell and what I've done, it is reconciling. You have these morphologies that do correlate with different cell types. So in a lot of the Deepcell original research shows the differences between these immune populations, differences between different cell types within a biopsy sample in different patients. But I think the real crux of the question is, how is it connecting to the genotype, and how is it connecting to these underlying things that sort of dictionary and that connection hasn't necessarily been possible until now. And so we're still building that data. We're still building those models out. But I'll definitely let Kelly if she has anything else.
Yeah, sure. That was a fantastic answer. I will say that, definitely we have seen that morphotype correlates with cell function. We haven't specifically characterized M1 versus M2, so I can't comment, with any empirical evidence on that. But we have compared, for example, activated versus naive T cells, which can be distinguished. We have compared monocytes versus macrophages, for example, which also can be distinguished. We've also studied different stimuli that are known to activate macrophages in specific manners. For example, oxidized LDL versus E. coli versus LPS, and where we know those mechanisms are similar, we see, for example, with E. coli or LPS, we see one specific morphotype or one clear reaction versus with LDL, where we know that there should be a different downstream sequelae of that activation, we see another distinct morphotype or morpholomic profile associated with that differential stimulation. So hopefully that helps.
Thank you very much. So when it comes to looking at morphotype, how do you view it as something that's transient, reversible, or do you view it as something that's more permanent?
It's a good question. It's a question we get often. Yeah, I mean, think of it as with single cell RNA transcriptome data. Um, it's affected by sample preparation, of course, but we think in the data we're getting out of it, it is more stable than our single cell RNA-seq data. So we've been looking at our cells that I've just presented with some of this pilot sequencing. And depending, it's more stable when you're shifting the protocol a bit from different person to different person. It's still affected, of course, because of the sample preparation, but it is more stable in our hands. And as I shown in the clonal information, the clonal morpholomic data, that you're having a distillation on a specific morphotype for these different clones. And so that has held up after more passages, albeit there's more heterogeneity. But as of right now, we are seeing that it's holding up.
Thank you very much. Next question: Can this technology be used to analyze images as opposed to cells? I guess single cells.
Wait, could you repeat that?
Sorry, I think, can this premise be used to analyze microscopy images, for example, or to look at uh, native histology or something like that?
Ah, yes. So just this general idea of using like AI and
Yes.
combining these morphometrics and everything. Yes. Yeah. So that's a lot of groups out there. I'm actually working with a group at MD Anderson, Dr. Yin Yuan. We're specifically focusing on the histology and understanding the morphologies in situ with H&E and some of these in situ sequencing and connecting it and trying to determine how morphology translates from tissue to dissociation. And so when we're doing that, we build custom models, and it's very similar. This approach of development of these quantitative AI models and integration with the different images is totally possible. Yeah, so you can do it. I've done similar things, not at the same scale as Deepcell, but similar things previously at UCSD of integrating different data and interpreting it and understanding how the morpholome changes.
Personal question I had: when you're analyzing cells using Deepcell, is it a 3D image or is everything being viewed from a singular plane?
Yeah, I get that question a lot too. Yes, so it is a single plane. So the question I get that's also a follow-up is, well, how does that change when you look at the different slices going through Z? So I worked with the group at UCSD that was doing imaging cytometry and computing it in waveform in 3D. It's extremely computationally intensive, and you don't get that data right away. But you're going to get more rich information, but there is a limit when it comes to that. We are reaching a limit of how much we can do, and information we can get with our images versus the time it takes to analyze and then make those sorting decisions, right? So even though it's a single image, it's a single plane, the resolution, it's like 60x and high micron resolution. We're able to get that information and just tell the difference between these morpholomic profiles, between samples using that information. And a lot of other groups have done something similar using these imaging cytometry data from like Amnis, and being able to look at that, and you still get it holding up.
Thank you very much. Is it possible to visualize the same cell over a period of time? Repeated sampling?
Uh, the same single cell in this
Yes,
system?
because a
No.
lot of times when we talk about morphology, it's almost, and this is maybe a technological limitation, but in the past it's always been almost watching a single cell migrate across a plate or something like that,
Yeah.
as opposed to looking at a population of cells, just because we didn't necessarily have the capacity to look at each individual cell morphology uh, one by one, like manually. S-
Yeah.
I'm curious if
With
this
this,
technology will allow the same thing.
I would say from what I'm thinking about it, and Kelly can correct me, but I would say no. Um, I think we need to think of this as a different experimental imaging modality, because, like, with traditional flow, yes, you're exposing these cells to different things, and it's going to affect the morphology. It's more gentle. Microfluidics are generally seen as more gentle. It's definitely has a little bit lower pressure on these cells from what I've seen, and they're a bit more happy. I've used microfluidic devices. I've used the regular image flow cytometry before, and you can see more differences, and it takes longer. Whereas the deep cell doesn't take that long. They recommend under 3 hours, but usually, to run your samples, it's like an hour. And so your cells are sitting on ice a shorter amount of time. They're not in suspension as much. But the idea that you want to observe a migrating cell and then sequence it, you can do that now even without deep cell, but you're limited on the human operator to actually get those cells out. So then how much are you imposing by heterogeneity and effect on morphology using a laser to cut it out? Or using trypsin and focusing and then putting that one cell into a well and then waiting to get 1,000 more if possible, if you have enough caffeine to get through that day, right? And so then you're putting stress on it as well. So I see is we're always stressing the cells out. Of course we're doing that. But when you take it and you say, if we're stressing the cells out in the same way, then we're able to look at this data and trust it, even though they may not, some of them may be adherent and we're putting them in suspension. What do you think we do with single cell sequencing? Of course. I think, the morpholomics, it's a lot of people are a bit harsher on it because it's new, but that's the same thing that happens with other single cell techniques.
So I think, yeah, I wasn't trying to be harsh about it, but I'm just kind of curious if you could, for example, capture like the halfway stage during a migrational process, because these are visible processes, so maybe it's just kind of a mental association with looking at something while it's in the middle of physically exerting an action, and then seeing if we can capture those intermediate steps. Like,
Ye- So, as a general question, yes, you can do that. Photo activation. There's some papers out there that if you have these wound healing assays, I forget what group it was out of Harvard. They created these photo activatable dyes in addition to some Genalia photo activatable dyes. So when you have the wound healing, they migrate it in, and then you photoactivate across the wound. You use traditional fluorescent cytometry, pull those out and sequence them. And it wasn't surprising, like the transcriptomic data from the single cell sequencing of those cells showed, EMT programs up and things like that, because you're only focusing on that area that's been invaded. Now, it's a little bit harder when you're trying to get these cells because there is information that's propagated through the back. So you have cells that get the cue to start moving, and then they moved, but then it propagates that information through the sheet at different ways and through different directionality and speeds. So then it's interesting to see the spatial temporal information for that. So then can you do these photo activations of those different areas? Yes. But now you're running into this issue of you have a few cells because there's more cells in this like 50 micron wide wound, but then you're getting to less and less cells, and that when you're going further and further into the sheet, you're getting different information of these different programs that are turned on, that you might find more insight than just focusing on the wound area. So you can do it now, but I think it's at a much lower throughput.
And I think related to that, I want to ask, how can you capture spatial interactions? The morphological properties, for example, of a cell attached to a basement membrane versus a cell not attached to a basement membrane.
Yeah, that is, I don't know. That is a good question. It would be really hard. I mean, you would have to do in situ like imaging, right? To take a sample, and then to take a biopsy sample, you would slice it and do like H&E or something, or even do things like a xenium. And then you can get some information of like which are detached, because then you can look at these MET programs that are turned on that are ones that aren't attached and ones that are attached. But when you do that, you don't get single cell resolution.
Mhm.
And a lot of these platforms are just based upon propagation from the segmented nucleus out in a polygonal way. So you're losing the morphology, the single cell morphology of it. And a lot of the other groups out there focus on patching. So if you look at the AI models that they develop for like pathomics, right, pathology-omics, they're looking at patching and not single cell data. And so there's still an opportunity there for us to get single cell data. And then you kind of can match it through, and dissociate those and then run it through Deepcell, because you can do these fixed cells as well. So there's live cell capabilities, but also fixed cell capabilities. Yes, the morphology is going to be different, but you know that going into it. So you're trying to understand how the morphology is different and why is it different, and how do you connect and create a dictionary between suspension and in situ.
Thank you very much. I think we have time for just one more question. I was curious, what's the best way to basically calibrate the deep cell system to detect maybe any errors, or to make sure that all the reads are, or the maps that being generated are accurate per se?
I'm not quite sure.
So, so basically, if you're if you run two populations of cells through the system
Okay.
and you're getting different results, is how do you ensure that the differences are differences between the two cell populations versus something else?
I see what you're saying. So it could be like preparation differences and culturing
Preparation,
differences.
imaging,
Yeah.
gating, all sorts of.
So you can save the gating. So when you create your classifier model, you can save the gating and pass it on to multiple experiments. That will give you insight on what has changed. Um, when it comes to the images, if you've seen the images that I showed, they all have similar contrast and background, because one of the parameters is focus and with the position and where it is to the objective. So you're getting them in the same position with the same focus and same background contrast, so it's not affecting the AI model to do that. So I'm confident in the image processing and image analysis portion of it. But when it comes to sample preparation, that's that's inherent to differences in just day-to-day heterogeneity, operator heterogeneity, and there's a lot of work that goes into battling and combating these intrinsic biases that we put in. And so I think just having, maybe the same operator, the same sample prep, and really standardizing it will allow you to figure out where in the process could there be changes, and is it a real change or is it not a real change?
Are there any hallmarks in the data where you could look at and go, "Oh, that's an artifact"?
The way I've seen some of the images that have been discarded is like maybe something out of focus, something not in frame, or cells that are going to be like it's not like extra. It kind of looks like an extracellular body, so it's a very very small. So then size. So you're able to do things like that, similar to what we do in microscopy, in traditional microscopy, and traditional imaging cytometries. You're able to see what these obvious aberrations are in the data, and then not use those images.
All right, thank you very much. Thank you. I want to extend very warm thank you to Andres, Courtney, and Kelly for this discussion, and we really do appreciate having all of you here today.
Well, thank you. I enjoyed it.
If
Thank you
anyone
very much.
If anyone has any further questions, please consider reaching out to our speaker directly with their contact information shown on the screen. As a reminder, this webinar will be archived on the scientist's website, and you will receive an email notifying you when the on-demand version becomes available for viewing. Please make sure to follow the scientist on social media for updates on future webinars and other news and events. In closing, I'd like to again thank everyone who took the time to join us today, and particularly those of you who did share your questions and comments. On behalf of the scientist, I'd like to thank our speaker, Andres Nevarez, as well as our sponsor, Deepcell. Thank you, everyone, and have a pleasant day.